All right, guys, we're going to start today with Chapter 1, an introduction and history of microbiology. So I like to start this course by asking you, why do you need to study microbiology? And I think in order to be able to answer that question, you really need to understand what is microbiology and what are microorganisms. So microorganisms are things that are generally too small to be seen by the unaided eye. And they include algae, protists, even small higher animals like worms, include fungi, viruses, and bacteria. And for the purposes of this course, we're going to focus primarily on bacteria. So, what is microbiology? Well, generally, it's the study of living things that are too small to be seen with the unaided eye. And a lot of people have a misconception that these germs are all bad. Well, in reality, 99% of bacteria are harmless, and some are even beneficial. Less than 1% of bacteria are actually harmful and cause disease. Microbes play a big role in our lives. They were once classified as plants, giving rise to the term flora for microbes. It's since been replaced with the word microbiota, but if you ever see flora or microflora, know that they're referring to bacteria or microbes. Microbes normally present in and on the human body are called normal microbiota. And these are our first line of defense against pathogens or disease-causing bacteria. Because of competitive exclusion, normal microbiota prevent the growth of pathogenic organisms. And their abundance can be quite surprising. On the surface of your skin, there are as many as 2 million microbes per square inch. In feces, there are 100 billion bacteria per gram. In soil, you might find as many as 10 million microbes per gram of healthy soil. And in the air we breathe, there are 50 to 500 microbes per cubic foot of air. Humans, both on and inside our body, have 100 trillion cells. But only 10% are our cells. 90 trillion of those cells are microbial. There are two different major groups of normal microbiota. We have resident microbiota, and these are the organisms that you'd expect to always find present, usually in the same relative abundances on your body. Transient microbiota are organisms that you'd pick up through regular exposure, and their variety are constantly changing. Developing your normal microbiota is something that happens almost immediately after birth. Within 12 hours of delivery, Staphylococci, Streptococci, and Lactobacilli will begin to colonize a newborn human. And if a mother breastfeeds baby, then the microbiota of baby's large intestine will consist mainly of bifidobacterium species. And major milestones in baby's life, like teething or eating your first solid foods, adding new foods to the diet, weaning from the bottle, these are all things that have significant impact on baby's microbiota. And it can take, up to a year of baby's life for normal microbiota to fully develop and establish. And this infant microbiota has a lot to do with why babies are so much more susceptible to disease than older individuals. All right, there are some parts of the human body, though, that should be microbe-free. And we'd expect microbe-free areas of the body to be like within your muscle tissue, your lower respiratory system, so trachea and bronchi should have very little microbiota. But as you go even smaller into the bronchioles and alveoli, there should be no microbiota, and it should be sterile. There should be no microbiota, and it should usually be sterile in the upper reproductive tract of females. The upper urinary tract, kidneys, ureters, bladder, those should all be free from bacteria. Your heart and blood vessels, your brain and spinal cord, muscular and skeletal systems, these should all also be sterile and free of bacteria. Liver, glands, bone marrow, and the middle and inner ear, internal eye, and sinuses should have no normal microbiota and are usually sterile as well. Now, opportunistic pathogens are organisms that are part of our normal microbiota, but under just the right circumstances, they can cause disease. So, compare that to a pathogen, which is an organism that always causes disease when it can. Infectious disease is when a pathogen overcomes the host's resistance and disease results. Emerging infectious diseases are new diseases or diseases increasing in incidence. Things like MRSA, AIDS, and E. coli 0157H7. Now, MRSA is methicillin-resistant Staphylococcus aureus. It causes a variety of infections. Penicillin resistance first developed in microbes in the 1950s, very shortly after the invention of penicillin. In the 1980s, methicillin resistance also shortly followed the introduction of methicillin. And in the 1990s, MRSA resistant to vancomycin was even reported. Escherichia coli 0157H7 is a toxin-producing strain of E. coli. So most E. coli are not pathogenic and don't make people sick. But this strain is. And it was first seen in 1982. It's the leading cause of bloody diarrhea worldwide. Acquired immunodeficiency syndrome, AIDS, is caused by the virus, human immunodeficiency virus, or HIV. So HIV is the virus that causes AIDS. It was first identified in 1981, and a worldwide epidemic followed, infecting 33 million people. There are 7,500 new infections every day. It's a sexually transmitted disease affecting both men and women. All right, reemerging diseases are the resurgence of old diseases due to extensive travel, a lax attitude towards vaccination, increased population with compromised immune systems. So the world is much more mobile today than it ever has been in human history. There's also an attitude among some people who've never encountered a lot of these diseases like polio or measles. Why vaccinate my child against these things if no one I know has ever had them? And increased population with compromised immune systems. Our medical care in the U.S. has become so great that we're keeping people alive that would not have been able to still be with us in decades past. So we have a larger susceptible population than we have when vaccination was taken more seriously. So diseases like this, reemerging diseases, include tuberculosis and pertussis. There have even been outbreaks of measles. All right, tuberculosis reemerging in the U.S. Tuberculosis is caused by Mycobacterium tuberculosis, and it causes infection in the lower respiratory system. Inhalation of airborne bacteria from infected people causes the disease, and 9 million people each year develop TB. There are 2 million deaths annually from tuberculosis, and there are about 2 billion people worldwide who are infected. 14,000 cases annually in the U.S. arise mostly from foreign-born individuals, but it's increasing at a rate of 2% each year. Pertussis is commonly called whooping cough, and is caused by Bordetella pertussis. And this causes an infection of the lower respiratory system. And before DTP vaccination, about 50% of people would develop pertussis. Vaccination significantly decreased this disease. In the 1980s, the number of cases had increased significantly, affecting adults and adolescents. So people who aren't up to date on their boosters were becoming infected. And immunity to the DTP vaccine declines after a few years. So if you get your childhood vaccination but no boosters, Usually by the time you're 12, you have no protection left from your childhood vaccinations. All right, let's move on to microbes in the environment. Microbes are really important decomposers. So both bacteria and fungi are really important for breaking down sewage and sewage treatment plants. And they can also even break down toxic waste like oil spills and petroleum products and other pollutants. Food spoilage is another. So lots and lots of food are lost each year to bread mold, soured milk, rotted fruits and veggies, canned food spoilage, and billions of pounds of food are lost each year. However, we can also capitalize on these microbes to help compost that food waste so that the nutrients in the spoiled food are available to be used to grow the next year's crop. All right, microbes also have an important role to play in element cycling. Carbon, nitrogen, and sulfur cycles are all dependent on microbial action. Photosynthesis and fixing carbon from the atmosphere, a lot of this is done not just by plants, but also by algae and cyanobacteria. Only certain microbes are able to fix atmospheric nitrogen so that other organisms can utilize this. Nitrogen is important for supporting the entire food chain. Without these nitrogen-fixing bacteria, we wouldn't have plants. We wouldn't have animals. So they're incredibly important. Where can nitrogen be found in our bodies? So here's a clue. Adenine, guanine, cytosine, and thymine. Which biological macromolecule is made up of these 4 nitrogenous bases? It's DNA. Good. All right, one more for you. What about this? Here's a picture of an amino acid. And there's that nitrogen down in the common amine group at the bottom. These amino acids are the building blocks of proteins. All right, microbes are also really important for food production. Things like alcoholic beverages, cheese, yogurt, bread, fermented foods like sauerkraut or pickles, and vinegar. And microorganisms themselves can be food, right? Like yeast and Vegemite or mushrooms. And believe it or not, there are actually some pretty huge advantages to studying microbiology. We can get results in the micro lab so fast, like hours or 1 to 2 days, and we get billions of cells per milliliter. Macrobiologists, vertebrate biologists would kill to have even thousands of individuals in their study. Billions is just unthought of. Microorganisms display the same fundamental and genetic properties that we see in vertebrates and other forms of life. So what's true for an elephant is also generally true for bacteria. And the space requirements for studying microbiology are minimal. We can get billions of bacteria in a space the size it would take to set your coffee cup down. It's relatively inexpensive, depending on the media you're using. All right, so let's transition here into a brief history of microbiology. Scientists searched for answers to basic biological questions. Is spontaneous generation of microbial life possible? What causes disease? And how can we prevent infection and disease? Events that led to the field of microbiology start with the debate over the theory of spontaneous generation. And Francesco Redi began to challenge this standing notion in 1668. In 1673, the microscope was invented and the microbial world was discovered by Anton van Leeuwenhoek. So the debate over spontaneous generation goes all the way back to Aristotle in the mid-300s BC. This is the hypothesis that, that organisms arise from non-living matter and some vital force forms life. Of course, the opposing theory to this is biogenesis, the hypothesis that living organisms arise only from pre-existing life. So Reddy conducted this famous experiment in 1668. He filled 3 jars with decaying meat. He left one open to the environment, one he sealed with a cork, and a third he covered with a fine mesh. Now, in the unsealed flask, we see on the rotting meat, flies have appeared and also maggots, and they're doing very well. In the sealed flask, however, no flies and no maggots are present. And in the flask covered with gauze, there are flies, but only outside the jar. And there are maggots on top of the gauze, but not inside the jar as well. And the maggots on top of the gauze are not doing well. They don't live long before they shrivel up and die. So this shows us that there's something different here than spontaneous generation. If all it took to create life was rotting meat, then you'd expect to find flies and maggots inside all 3 jars. All right, the first evidence for microbial life came in the 1673. So Anton van Leeuwenhoek is commonly referred to as the father of microbiology because of his discoveries. He developed the first simple microscopes, and he examined water and saw tiny animals, fungi, algae, and single-celled protozoa. He called these organisms animalcules or beasties. In 1745, John Needham did a follow-up experiment where he boiled nutrient broth, think of something like chicken broth, and he boiled it in covered flasks. Put boiled nutrient broth into covered flasks. Okay, nutrient broth was heated and then placed in a sealed flask. Boiling was thought by some to kill all life. However, he saw turbid microbial growth. In 1765, Lazzaro Spallanzani criticized Needham's study, suggesting that microbes from the air had entered his flasks. Spallanzani boiled nutrient solutions in flasks and then sealed them immediately. Critics said that air is needed for life. So nutrient broth placed in a flask that's heated and then sealed immediately. And there was no microbial growth. The solution remained clear. In 1861, Louis Pasteur demonstrated that microbes are present in the air, and this helped to debunk spontaneous generation. Pasteur's experiments, he used a swan-necked flask, and when the flask remained upright, no microbial growth. So this flask is shaped like a swan, where it's got like a round body, but then it has this neck that has a dip in it, kind of like a teapot. And if the flask was upright, there would be no bacterial growth. But if the flask was tilted, dust from the bend in the neck would seep into the flask and make the infusion or the broth cloudy with microbes within a day. Perfect. So you can see in step 1, Pasteur poured a beef broth into a long-necked flask. Microbes were present in the broth, right? Next, he heated the neck of the flask and bent it into an S shape. Then he boiled the broth for several minutes. Microbes were not present in the broth after boiling. And then the third step, microbes did not appear in the cooled solution even after long periods of time. So it remained sterile. All right, so how does the scientific method fit into all this? Spontaneous generation debate led to the development of the scientific method. Observations lead us to questions. We use our questions to generate hypotheses and make predictions. And these hypotheses are tested through experimentation. Results either support or disprove our hypothesis. We either accept our hypothesis, and over time that can lead to the development of theory or law, or we reject our hypothesis and modify them to get closer to the truth. So, what does cause disease? Well, Louis Pasteur developed the germ theory of disease. It's microbes that can cause disease. And Robert Koch took this a step further, and he studied causative agents of disease and developed experimental steps for directly linking a specific microbe to a specific disease. And he was able to demonstrate that anthrax was caused by Bacillus anthracis. So Koch's postulates, or his kind of rules for connecting a microbe with a disease, are that the suspected microbe must be found in every case of the disease, but be absent from healthy hosts. The suspected microbe must be isolated and grown outside the host. And when the microbe is introduced into a healthy, susceptible host, the host must acquire the disease. So the microbe has to cause disease. And the same microbe must be found in the diseased experimental host. Koch's work led to vaccine development and the eradication of smallpox. Smallpox is the only disease humans have managed to eradicate from the planet. I think the only samples left are in a couple governmental deep freezer labs. So no one has to be vaccinated against smallpox anymore because nobody gets it. It's gone. It's extinct. We've also seen with the use of vaccines significant decline in polio, measles, and other childhood diseases. Koch's work has also led to the development of antibiotics. So how can we prevent disease? Well, in the 1840s, Ignaz Semmelweis made the connection between disease spread and hand washing. He hypothesized that medical students were transferring cadaver particles from autopsy studies into delivery rooms. Mothers would develop puerperal fever. Now we know puerperal fever is caused by a bacterium in the genus Streptococcus. It began requiring medical students to wash their hands with chlorinated lime water. And mortality in the subsequent year dropped from 18% to 1%. Unsuccessful in gaining support for his method, he later developed depression and was committed to a mental hospital, and ironically died of a streptococcus infection. So what do you think? If you or someone you love were having a baby, would you want their doctor to be washing their hands? All right, Joseph Lister picked up the mantle and continued to work on this question of how can we prevent the spread of disease. Lister is stated to have saved more lives in the 19th century than all the wars during that time sacrificed, and he introduced a system to prevent surgical wound infection. The system is aseptic technique, techniques used to prevent contamination by unwanted microorganisms. We'll be working in the lab to get you all familiar with aseptic technique and how to use it appropriately. All right, microscopy. So 3 factors determine the quality of an optical image: the magnification, the resolution, and the contrast. Magnification, using our compound microscope, is the 2-lens system. The objective lens is in the nose piece, and that's the one that you'll turn on the microscope to select. There's also a lens in the ocular, or the eyepiece. So the total magnification of what you're looking at is the magnification of the objective times the magnification in the ocular. So for our microscopes, the ocular lens is a 10 times magnification. The objective you'll choose. So let's talk about the metric unit and units of length. We're all familiar with meters and centimeters, right? There's 100 centimeters in a meter. You all know there are 1,000 millimeters in a meter. But in this class, we're going to be going even smaller than that. A micrometer, or a micron, is 1/1,000,000th of a meter. And that's about the diameter of a white blood cell in humans. Nanometers are 1/1,000,000,000th of a meter. And this is something more on the size of the diameter of a virus particle. All right, so when we're looking at a sample, there's light moving through the air, and then when it hits the glass, it's refracted. Now, lenses take advantage of this refraction to invert, reverse, and enlarge an image. So see how the specimen on the left has the flagellum or the tail at the very bottom, but in the image you see through your microscope, the flagellum will be at the top. It's inverted, reversed, and enlarged. Now look at the red eye spot there. It's on the bottom left corner of the specimen, but under the microscope it'll appear at the top right. All right, resolution is our ability to separate two objects that are close together so they're seen as two separate things. So on the left, we can resolve that there's different clumps of bacteria there, whereas on the right, it just looks like one big blob. All right, so your microscopes, you have a number of different objectives. The most powerful objectives, you'll need to use immersion oil. So if you look on the left, you'll see a sample on a microscope being observed without immersion oil. So you'll use this for your lower power objectives. Light will move through the glass cover slip, through the air, and then up into the microscope objective. When we use immersion oil, it helps keep more of the light focused on the microscope objective by preventing the light from passing through air. And we'll use immersion oil to fill that gap when we look at our highest power resolutions. All right, the next property you'll be looking at on the microscope is contrast. This is differences in intensity between two objects or between an object and a background, right? So a clear microbe in water on a glass slide is going to be really difficult to see. Whereas if we were to stain that clear microbe with a purple stain, they'd be much more visible, right? So by increasing the contrast, we can make it easier to observe things. So on the left, we see bacteria that are unstained, whereas on the right, same type of bacteria, but a stain's been applied. And it's much easier to see the purple stained bacteria. All right. And these compound microscopes we have in the lab are not the only types of microscopes available. So we'll be using light microscopes. They have bright fields, they're most common and easy to use, and they're one of the most important tools for studying living microorganisms. And they can magnify things up to 1,000 times using the oil immersion lens. An electron microscope can magnify things in excess of 100,000 times. It's useful for observing the fine details of cell surfaces and internal and external structures. All right, here's some parts of the microscope. There's the ocular lens at the top, the eyepiece, and that's usually a 10x magnification. The stage where you'll place your specimen. The condenser under the stage focuses the light. There's an iris diaphragm that controls the amount of light that enters the objective lens. The objective lens itself is a selection of lens options that provide different magnifications. And the total magnification is the product of the magnifying power of the ocular times the objective. At the very bottom, there's the light source with the means to control the amount of light. And you also have a knob to control the intensity of light. All right, another type of microscopy is transmission electron microscopy. It's very high resolution and it's used to study the internal structures of cells. Scanning electron microscopy creates high resolution images of surface structures. And here's a picture of an electron microscope and a transmission electron microscope image. And now here are some scanning electron microscope images. Microscopy techniques include dyes and staining, and the purpose is to increase the contrast and resolution between microbe and background, and it makes the microbes more visible. Microbes are mostly transparent on a slide. We'll be doing a few different types of staining in this course. A simple stain is when just one stain or dye is used to provide contrast. We'll also apply differential stains, where we use two or more stains or dyes to provide contrast between different types of microbes. And we also have some special stains for staining certain cell structures or the background. So here's an example of a simple stain, a single dye used to stain cells. Some examples are methylene blue, crystal violet, or safranin. Now, differential stains are a staining procedure to distinguish one group of bacteria from another. We'll be doing gram stains, acid-fast stains are another differential stain, and endospore stains. So here's gram-positive organisms are purple, gram-positives are pink. And acid-fast stains are used to stain organisms of the genome Mycobacterium, which don't readily take up dyes or stains. They contain a waxy mycolic acid in cell walls, which resist the staining. And an endospore stain is used to stain cells which form endospores. Here's an example of a capsule stain. You see the capsule is kind of like a halo around the organisms. A flagella stain is used to stain the flagella of cells, which are normally too thin to see. All right, so that wraps it up for the introduction and history of microbiology. Thanks for listening, guys.